Good morning and welcome to the second day of the Summit on the Neuroscience of Twice Exceptionality. I'm Emily Langendorf and I'll be moderating this session. For anyone who's joining us for the first time this morning, please note the Ask a Question feature to the left of the chat to submit your questions during the presentation. We'll go through as many as we have time for at the end. And now I'll hand it off to Tomas to get us started.
Thank you very much, Emily. It's my privilege to introduce the first speaker of the day, Jake Michaelson. Jake started his career with a master in biological engineering at Utah State University and moved from there to the Technical University Dresden in Germany, where he obtained not only his PhD in computational biology, but also his impeccable German, I have to say that. He moved from there as a postdoctoral fellow to Jonathan Sabat's laboratory, and from there to the University of Iowa, where he right now applies a broad range of cutting-edge computational biology techniques to uncover the basis of psychiatric and neurodevelopmental disorders. It's my pleasure to listen to your talk today, and thanks for speaking here, Jake.
Thank you, Tomas, for that wonderful and kind and flattering introduction. It's great to have you as a colleague and great to have you be the one to introduce me. I really enjoy working with you. Welcome, everybody, and good morning. And I'm thrilled to be able to have the opportunity to speak to you today about some thoughts that I have about twice exceptionality, especially as it pertains to research in this area. And so I hope that you, as we go through these things, that you'll quite liberally put questions in the question box, because I hope this leads to some productive discussion. So I retitled at the last minute my talk, or the subtitle is "The Plural of Anecdote is Data." And that's a quote that's been tossed around several times in our collaboration. And I think it sets up a nice continuum between more qualitative research on one end and then more quantitative research on the other end. And in the first few slides of this talk, I want to talk about how we can effectively navigate this transition from anecdote to data. So anecdote might be recalling that you knew a kid one time that had these features, and isn't that interesting? And then data is, of course, that multiplied many times over again to the point that you can actually generalize in a productive way from that. But it's not just about amassing more data as we make that transition. It's about thinking about how to standardize and streamline the data collection so that we can achieve the large sample sizes necessary to gain deep insight into these phenomena. And then also thinking about what biases might be intrinsic in what we're doing and how we're approaching this problem. I'm putting up here an image of an airplane from a study that was done during World War II. They wanted to know where they should reinforce the bombers that were coming back. And so they kept meticulous data on where the holes were in the bombers that came back, with the idea being that those would be the ones that you'd want to reinforce. But you'll notice that there are some noticeable gaps in where those are. And of course, this is a textbook example, quite literally, of ascertainment bias or survivorship bias, where actually the places you want to reinforce are the ones where you're not seeing any dots, because those are the bombers that didn't make it back. So that's just an example of the kinds of biases that I want us to think about. And over the next couple of slides, I'm going to give some lighthearted examples of what we in machine learning call overfitting, but also more generally just making drawing conclusions that are too strong for the data in front of us to support. So the first example I thought of is the game Clue, which is a game that I'm sure many of us have played before. But if you haven't, the way that it works is you try and find out who the murderer is. And every player has certain evidence, and throughout the game, you move around the board, and you interact with the other players, and you assemble evidence so that by process of elimination, you can figure out who the murderer was, what the weapon was that they used, and where they did it. And so drawing strong conclusions from weak data would be like if you, at the beginning of the game, had eliminated two data points, and then you were ready to make an accusation. which is kind of the climax of the game, where you feel certain enough that you can make an accusation. But the trick is, if you make an accusation and it's not true, then you lose and you're out of the game. And so that's just my first example of, and I hope we can align our minds to how this works in science and in research. The next one is a comic that some of you might know called XKCD. It's a web comic that's kind of very much about geek culture. And I'm not going to show you the whole thing, but I encourage you to look it up because it's quite clever. But this one is titled "The Problem with Statements Like No Party Candidate has won the election without state," or "No president has been reelected under blank circumstances." So it starts off in 1788, where the first statement is simply, "No one has been elected president before," but Washington was, of course. And so you have one data point at that point. And so the next time around you say, well, no incumbent has ever been reelected until Washington. And then you go on, and then it gets kind of funny. "No one without false teeth has become president. No challenger has beaten an incumbent." And so every time when you accumulate one more data point, you come up with one more way to thread the needle, or one more way to gerrymander the data, so that you can arrive at some specific conclusion. Of course, this ends up in more recent history with something as funny as, "No Democrat incumbent without combat experience has beaten somebody whose first name is worth more in Scrabble. That is, of course, until Bill beat Bob," and so on and so forth. So yet another example of drawing strong conclusions when we have very limited data in front of us. Now, the next one that I want to show you isn't quite as lighthearted. It's definitely much more serious. And we're getting more into the science now. Okay, so this is, of course, Andrew Wakefield, who many of you know as being largely responsible for the myth that vaccines cause autism. And there were numerous elements of misconduct associated with this work, and things that led to false conclusions. But I'm going to set all that aside for now, because that's not what I want to talk about. What I do want to talk about is that this study, even if it had been done completely honestly and transparently, was only based on 12 individuals. And they drew very strong conclusions, namely that vaccines were responsible for causing autism. And that has led to a culture of suspicion about vaccines, and has unfortunately led to the resurgence of deadly diseases like the measles. So again, another example of overfitting to a very small data set, and that's not even taking into account the biases and the falsehoods that were in the data. So before I go on, there are two facts that you need to know right now anytime I bring up Andrew Wakefield. And the first is that autism is overwhelmingly genetic. So there was recently a study of over 2 million participants and spread over multiple countries. And they consistently found that the heritability of autism is in the range of 80 to 85%. So largely genetic factors contributing to liability for autism. So we know that it's genetic, but we're still obviously, as geneticists, employed here, and that is because we don't know all the genes and mechanisms, and that's what we're trying to figure out. Second, that vaccines do not cause autism. So this has been looked at many, many times in incredibly large epidemiological studies that are very well powered to detect such an effect if it truly existed. And the latest being a study of over 650,000 individuals, and was unable to find a link despite that overwhelming statistical power. So how can we fix this problem? As I reread the Andrew Wakefield paper, which was published in 1998, and in a very prestigious journal, by the way, The Lancet, I was struck at how little detail was actually in the paper. You could not get away with that today. Editors and journals and reviewers demand much more thorough detail. In a paper I'm going to show you later, not even in the main text, but in the supplementary materials, which is something that's not part of the main text, had 168 pages of supplementary materials describing the methods and the data. So there's much more detail required now. More journals and funding agencies demand that the raw data be published along with the paper, and that's to facilitate reuse of the data, which is a great thing, but also replication and scrutiny, making sure that the methods and the data are what they say they are. And thirdly, many journals require the release of documented code that reproduces the complete analysis that is presented in the paper. But the question is all of this enough? Can you still draw strong conclusions from small studies, even if they follow all these guidelines? And I was thinking about how to present this, and I think there's a little bit of a dichotomy here in study size. And I by no means want to criticize small studies or their value, because I think they definitely do have a place. But limited sample size has been one of the #1 contributors to many of the reproducibility crises that we see, especially in social sciences and in psychology. And so there's waiting room size studies. And what I mean by that is if you could fit your whole sample that you're presenting in the paper in a waiting room, that's a waiting room size study. And these often suffer from low reproducibility. There's a high potential for bias because there's much more of a potential for cherry picking, even if it's unconscious, even if the primary investigator is not intending to do that. There may be ways that they're stacking the deck that they're not even aware of. And then finally, low power, the inability to have strong statistical associations. Now, you contrast that with stadium-sized studies where there's higher reproducibility, not to say that it's perfect. There's a lower potential for bias because there's so many more people involved, both on the participant side, but also on the investigator side. So there are many eyes on what's happening, and it's much harder to be dishonest if you're working in a highly collaborative environment. There's higher power, and as we'll see later today, there's the ability to see structure or groups in the data. So again, the waiting room size study, often the recruitment is done directly by the investigator or the scientist, the researcher. But for the stadium-sized studies, there needs to be recruitment by a collaborative network that is a combination often of clinicians or practitioners and scientists, all working together in a network, often international or nationwide, to recruit people at multiple sites. And so I want you to imagine yourself fitting into this framework, because it's highly likely that someday you'll be invited to participate in such a network. So some of the stadium-sized studies that are out there are, and I'm just putting down a smattering, ABCD, which is a longitudinal neuroimaging and genetic study of behavior in adolescents. There's the UK Biobank, which is largely older adults. All of Us is a somewhat newer initiative in the United States to recruit 1 million individuals to share information about their health and their lifestyle and their biology. And then finally, Spark, which I'm going to talk about in the next few slides, is the largest genetic study of autism. And I'm the principal investigator for Spark in Iowa, the site PI. So Spark, as I said, has over 250,000 participants nationwide. And this is a map depicting the density of participants, which tracks largely with population density. And we want to understand the experiences and the biology of autism, and with the hope that this knowledge will improve the lives of those with autism. So your stories or your student stories or those that you work with can be a part of our overall understanding of autism. And that is the message that I want to convey, is that by joining Spark, you become part of that embodiment of knowledge. So you can check out Spark by going to sparkforautism.org/UIowa. And again, I mentioned that there are parallel stories, the stories that your genes tell, and that influences your experience with autism. And as I mentioned, the heritability of autism is in the neighborhood of 80%. So there's a strong link between those two stories. And the great thing is that by understanding both of these and how they, what the interplay is between them, we have an improved understanding of how the brain works. Not just the autistic brain, but I'll say that actually a large chunk of human-centric neuroscience, and not even just human-centric neuroscience. We heard yesterday from Ted Abel, who does a lot of work in mice. But a lot of our basic understanding of how the brain works, of how neurons work, is fueled by funding for autism research so that we can understand how the brain works in the context of autism and in typical development. So the way that this is operationalized is that if you sign up for Spark, you get a kit in the mail that has a tube in it. You spit in the tube, you send that tube back to a lab, and that DNA that's in that saliva gets sequenced. And that sequencing data gets put in a database that is available to qualified researchers to ask scientifically tractable questions. In parallel with that, your experience with autism is captured by taking surveys online. So the thing that I want to emphasize is that your entire participation in Spark can be done entirely within your home. You don't have to go into a lab to be poked and prodded and to participate in this. So it's the barrier for entry is very low. Some of the benefits of participating in Spark is, I think the number one is that when scientists and doctors talk about autism, and also obviously educators and other practitioners talk about autism, they will talk about autism and you will be embedded in that because you will be a part of that overall fabric, that overall landscape that describes what the map of autism is. And there's also access to high quality resources like those at the Discover page on Spark. There's up to $75 of initial compensation. There's additional compensation for participation in subsequent studies as well. There's the potential for return of genetic findings with genetic counseling at no cost to you. And currently, we're quite conservative with the results that are returned because we only want to return results that we're very certain of. And that translates to about 10% of Spark participants receiving a genetic finding. And then also there's the opportunity to participate in follow-up studies that are specifically relevant to your situation. So some of the things that we're learning from Spark are depicted here, but the thing that I wanted to emphasize was that Spark has launched over 100 follow-up studies that seek to better understand different aspects of autism. So some of those include, and I'm cherry picking here because many of these are things that we have performed research match studies in, and you'll hear about some of these later today. So sleep issues, eating issues, language ability, sensory issues, and then abilities and strengths, sex and gender identity, which is a research match that we recently completed, and then also treatments that are tailored to the individual. And then finally, for this part, I just wanted to put up the list of what we call Spark genes, or these are the lists of genes and parts of the genome that have been robustly linked to autism. So you may look at this and think that this seems like a long gene, but in reality, we expect, based on forecasting and projecting, that the list may be 3 to 5 times longer than this. And but some of the common themes among autism genes are that they tend to be expressed in the brain. They tend to be expressed during fetal development. Many of them, as Ted Abel mentioned yesterday, are involved in gene regulation. And many others are involved in the neuronal synapse structure and function. And so the working hypothesis, which is a very broad one, says that the flavor of autism that one presents with depends on the combination of genes that are affected in that individual. And we think that very few individuals can attribute the autism specifically to a mutation in a single gene, but that more often than not, it's a combination of numerous genes that are affected in different ways that together affect neurodevelopment and result in a state of mind and behavior that meets criteria for autism. So what can you do? Because I want you to insert yourself into all this, even if you're not a researcher. So a few suggestions. In general, I hope that you will commit yourself to advocate for research. There are a number of different ways that you can do that, to be aware of the research opportunities that are around you, both local and online. To make research participation a part of your culture. You should ask yourself, I cannot answer this for you, but I hope that you'll ask the question, do I have a moral obligation to participate in research, or to at least advocate for research participation? So here I've shown, I put up a picture. I had to dig through my files to find this, but this is a picture of me as a postdoc. And I had an epiphany one day that I was a little bit frustrated that I was working with data from all these studies, so many human participants, and I had never participated in a study. And so I resolved that I was not going to let that go on any longer, and I volunteered for a study. And since that time, I try to volunteer for any study where I can be of use. An example of that is that I know, because I've had my genome sequenced, that I have a non-functional LDL receptor gene. And so what this results in is astronomical cholesterol, not just like, oh, that's a bad lifestyle cholesterol, but like really off the charts cholesterol. It's managed, thankfully. But I do participate in clinical trials that are looking at familial hypercholesterolemia. And I see that as kind of a moral obligation, because I know that I have something unique about myself that requires study, and that I can volunteer to contribute to that. And so I think it's incumbent on us to promote a culture of not just informed consent, but informed and motivated consent. That doesn't mean that a person has to participate in every study that comes their way. There may be reasons why they choose not to. Maybe there are risks involved in participating in that study. But largely those risks are filtered through an ethics panel called an institutional review board. And there are typically, measures in place. And so I would encourage you to have that paradigm shift of being kind of opted in by default into research, and then also to be ready when researchers call, because we will call. And then once you do participate, be aware that results do take time. Research is often a glacially paced process. So be patient, but I do enjoy hearing from participants every now and then who are just checking in and want to see if we've found anything new. Finally, there are a couple of websites I put at the bottom there that if you're interested, you can navigate around them and look at what studies are available. You can sign up to be notified if you qualify for a study. And then more specifically, I'm putting up the Spark URL again to please go and learn about Spark and email us with your questions. And here I have a picture of my son, Jethro, because I tried to pass this on to the next generation, this idea of being eager to participate in research. And so he's holding a 3D print of his brain that he got because he had his brain scanned in an MRI study that he participated in here at the University of Iowa. So that's all kind of the more philosophical big picture part of the talk, and I'm going to get into some data now in two parts. The first part is that I'm going to focus on cognitive archetypes in high ability students, and we're going to ask how they relate to autism. And then secondly, for the last part of my talk, we're going to look at creative archetypes. And what are those creative archetypes that we can observe, and how do they relate to behavior, and how do they relate to genes? So by now you've heard a couple of talks on our collaboration, but I just want to put up that picture there again and thank my collaborators, Susan and Thomas and Ted, for a very enjoyable and edifying collaboration to be a part of. I think you also heard that one of the first things that we did together in our collaboration is we received pilot funding from the Simons Foundation to digitize the clinical records of the Belin-Blank Center, which were paper records. And so it was a mammoth task to design the database, you got to physically separate and scan and enter the scores from over 1,000 clinical records. And each file took about 30 to 60 minutes to process. And so the final size of the data was 1,074 unique individuals over 1,200 variables. And I just wanted to put up a picture of Lucas Caston here, pictured with me at NAGC in 2019, who is an excellent research assistant, and now he's a graduate student in the genetics program in our lab. But he played a big part of that. Obviously, that took the work of a lot of people, but he was one of the main organizers of that effort, and so we're really grateful for him. So the scientific premise, at least part of it, is that almost universally high IQ is seen as a protective factor, whether you're talking about neurodevelopmental, neuropsychiatric, or neurodegenerative disorders. And so one thing that we were just curious about is whether there were any domains or contexts where high IQ might actually be a liability. And some of the real world justification for this focus are that, as we've heard, twice exceptional individuals have substantial contributions to make to society, but they're blocked by behavioral and other challenges that they face. And the other is that many interventions are designed with lower functioning individuals in mind, and they don't work with 2E individuals. So somebody asked about this yesterday, and I wanted to make sure that I put it in, but this is the overall distribution of individuals in this data set according to their diagnostic status. And then as you can see up here, this is stratified by whether or not they have a full scale IQ of greater than 120 or not, which is one of the how we kind of operationalize the gifted variable that we were looking at. And so what I just want to draw your attention to is that, as was said, autism actually makes up a smaller chunk. Now, this doesn't sum up to the total number because, of course, people can have multiple diagnoses across these. But we have 126 individuals with autism in this data set, and 369 have no diagnosis. And so this is generally the breakdown of the various clinical diagnoses that are seen in this sample. But here we're going to focus on 2E with ASD. I'm not going to talk about what is IQ. This was actually from a different talk that I was giving to more of a lay audience, to people who might not, think a lot about what IQ is. But I can skip over this because I know I can safely assume that 99+% of you have a good grasp on what IQ is. But this is the overall flow of how this works. So we have, again, the paper records from 1,074 gifted and talented children, or not. They're actually not all children. But they were scanned and digitized, and we put them in a REDCap database. And then our analysis kind of forked off into 2 arms. We have a diagnosis-focused analysis, where the main unit that we're looking at is we're looking for ASD diagnosis by IQ interactions. In other words, trends that differ according to IQ, but are contextualized by whether or not you have an ASD diagnosis. And then on the bottom one, we looked at some cognition-focused analyses, where we looked at the index scores from IQ tests, and we constructed cognitive archetypes and looked at what those archetypes were associated with. And then in the end, we kind of bring it all back together to synthesize it together. So for the first approach, we took autism diagnosis as one variable, and then we took full-scale IQ as another variable. And so together, what behaviors and academic patterns do these variables predict? So there's a main effect, which means that the effect is independent of diagnosis. And then there's an interaction, which means that the effect depends on the diagnosis. And so for the statistically inclined among us, this is approximately the model. We have some covariates in there, but these are the main features that we're looking at to see if diagnosis and IQ and the interaction between them have substantial explanatory power on whatever Y is. And we kind of cycle through different achievement and behavioral variables. So just 2 examples of this. And again, we're focused, there are significant main effects, but I'm not going to focus on those, because what I'm particularly curious about is where there's an interaction between these 2. And in other words, as you can see here in gray is the trend of those who do not have an ASD diagnosis. They may have some other diagnosis, but they don't have ASD. And then in red are those with an ASD diagnosis. So you can see here that for this interpersonal relationships score, as IQ increases, if you don't have ASD, then generally that interpersonal relationship score gets better. So higher IQ, better interpersonal relationships. That trend is actually the opposite in individuals with an autism diagnosis, meaning that if you have an autism diagnosis, as you increase IQ, the probability of you scoring high on this interpersonal relationship score drops as full-scale IQ increases. Similarly, if we look at a measure of anxiety, we see that if you don't have an ASD diagnosis, as IQ increases, your score on the anxiety scale goes down. But the opposite is true if you do have autism. So your score tends to increase with higher IQ. So we looked at these over a range of different variables so that we could understand how IQ has differential effects depending on whether or not you have an autism diagnosis. And this is a summary of, I don't expect you to really look through all of this, but I just want to show you some of the variables that ended up having a significant interaction. And what's color-coded here is red means that there's a positive slope for ASD and or either variable really, and blue means there's a negative slope. So for instance, let's see, let's pick the appropriateness of social responses score. So in general, if you don't have ASD, you go down on that score, which is kind of an indicator of a problem. And then if you do have ASD, then it goes up. And same thing with like the reciprocal conversation and some of these other ones. Anxiety is probably the most reproducible one that we see across various measures. Is anxiety is a big one. This is looking at it in a slightly different way, but these are other interactions where we discretize the groups, meaning that we cross the diagnosis variable with a binary indicator of whether or not you had an IQ of greater than 120. And these are parent-reported problems. And then here you see the high ability in ASD group. And then the red indicates the number or the proportion of the total where there's a problem indicated. So interrupting, attention problems, following directions, impulsive, school refusal, cannot play quietly, hard time waiting turn, and concentration. So these are all things that are, by the parent's account, more pronounced in kids who are 2E than even their normal IQ range, but ASD counterparts. Probably most worryingly is this endorsement of suicidal thoughts. This was the parent-reported problem that was most significantly associated with 2E ASD in this analysis. And so there was about a 4-fold increase in the rate of endorsement here among high ability and ASD, which was statistically significant. Okay, so that's our approach 1. Approach 2 now is looking at cognitive archetypes. So what if we ignore diagnosis and just look for recurrent patterns of cognitive strengths and weaknesses? And we could ask what diagnoses and what behaviors are these cognitive archetypes associated with? And we can ask, do any of these patterns suggest a testable neurobiological hypothesis? So before I get into all that, I just want to talk a little bit about this approach that we use called archetypal analysis. Now, if you work with data, chances are you have run into what are called clustering approaches. And these are basically ways, algorithms, techniques to infer a grouping structure within data, usually large data sets, without knowing a priori what that grouping structure is. So you can look at the relationships between different variables and identify that there are patterns that kind of separate out into groups. Some of the problems, though, associated with clustering analyses are that you have to choose, you have to come up with some way to choose how many groups there will be. I really apologize, I'm quite parched this morning. And then secondly, in most clustering techniques, there are a few exceptions, but in many clustering techniques, a person can be in one and only one cluster, which limits the usefulness of that if a person can only belong to one cluster. And then if they can be in more than one cluster, the question arises, how much more in one versus the other? And a lot of times what we found in our work repeatedly is that the results that you get, the groupings are just really not often meaningfully different from each other, which is, of course, one of the reasons that you do this. But an approach that we like to use that's a little bit of an alternative to this is, rather than looking for example, or rather than looking for cluster centers, which is what is done in these other clustering approaches, if you looked at extreme examples in the data, and in data science, the bubble that encloses a multidimensional data set is called the convex hull, which you can see depicted here. It's basically, if you, in like 2 dimensions, if you imagine a rubber band kind of wrapping around the extremes of your data, that's what it would be like. And then there's a generalization in more dimensions. And so a mentor, a scientist, a statistician that I was lucky enough to have as one of my mentors when I was an undergraduate, this is Adele Cutler. She's at Utah State University, and she is a very formidable statistician. And this is an approach that she presented together with Leo Breiman. And so the idea is to find K pure examples or archetypes. And these are points along the convex hull, of which all observations are a convex combination, meaning that you give every individual in your data set some coefficient between 0 and 1. And together, over the number of archetypes that you choose, those coefficients sum to 1, meaning that every individual is a mixture of the essential archetypes that you define. And so maybe in a more practical sense, you can think about it like this. Everything in our universe is some combination of these elements. So you could think of these elements as like archetypes, because it doesn't get more elemental than these. And everything that we see around us is some combination of these things. Or if you're into Carl Jung, and Carl Jung came up with this system of archetypes to describe characters and motivations and that sort of thing. But these are examples of archetypes. And so this is both from a numerical standpoint, but also from like a conceptual standpoint, what we're trying to achieve in this data. So the last thing I'll show you before I show you the results of this analysis, again, that we did on the index scores and other subtests of IQ tests, is that we have to choose the number of archetypes that we're going to settle on. And so the way that you typically do this is that you run a range of different possible numbers of archetypes. And at each stage, you examine the reconstruction error. In other words, how closely can I approximate the original data by choosing some number of archetypes? And this is an elbow plot. And what you do is you try to find the point at which there's a kink. In other words, at the point at which there's kind of diminishing returns between how complex of a model you're specifying and how well it describes the data. So of course, your ability to recapitulate the data improves over time, but you're having to add more and more archetypes, which has a negative effect on interpretability. So this is every line, every row here is an individual in the data set. And the archetypal analysis organized the data as you see here. So archetype 1, which we call A1, has generally high full-scale IQ and is actually pretty high across the board. A2 has relatively lower full-scale IQ, not so strong in processing speed, definitely not very strong in verbal comprehension, but a very specific strength in perceptual reasoning and lower on working memory. So that's another archetype, is A2. A3 is kind of low across the board. And then A4 and A5 are where it gets interesting. A4, you have this pattern of kind of middle of the road to higher full-scale IQ, but with a very profound strength in processing speed, a specific deficit in verbal comprehension, and then kind of so-so in perceptual reasoning and working memory. And then in A5, you have kind of the mirror image of A4, where you have a profound deficit in processing speed and a comparative strength in verbal comprehension compared to what A4 has. Now, the thing that I want to draw your attention to is that over here, we show within each one of these archetypes the depletion or enrichment of ASD cases, of ASD diagnoses. And what you'll see if you can squint your eyes a little bit here is that A5, archetype 5, is strongly and significantly enriched, 2.5-fold enriched for ASD cases, while A4 is depleted. The effects, like these changes are symmetrical, or they're not symmetrical, but they're centered at 1, and so a value less than 1 is depleted. And if you put this on the log scale, the effect is about the same, but in the opposite direction is this enrichment, right? So about a 2.5-fold depletion right here of ASD cases and a 2.5-fold enrichment here of cases. So the fact that these patterns are mirror images of each other and that they have opposite direction of enrichment versus depletion of ASD cases suggests to us that there's something really interesting going on here. And somehow it's a link between processing speed and verbal comprehension. And over here, you'll see some of the things that where A5 and A4 differ. So we basically take the difference between these two archetype scores and ask what they are associated with here. So unsurprisingly, fluency in almost any subject, whether it be math or reading or any other kind of fluency, is severely impaired in A4 compared to A5, and attention problems and lots of other things. The one thing that I'll point out that's very interesting is that reading comprehension apparently is improved versus A5 and A4. So the hypothesis that this suggests to us that we would like to test is that, and this was kind of alluded to in several talks yesterday, so I think this is consistent with other theoretical frameworks that are out there, but you can imagine that in our archetype 5, which is the low processing speed and high verbal comprehension, that you might expect weak inter-hemisphere connectivity and potentially stronger intra-hemisphere connectivity, and you might expect the opposite in archetype 4. And so this would be like a high risk for autism archetype, and this would be a low risk for autism archetype. And we have some preliminary data that suggests that there might be something to this, because we looked at genes that are known to be involved in the development of the corpus callosum. And Tomas mentioned the corpus callosum yesterday and presented some very interesting results there. And we would expect that on that interconnect between the hemispheres, that something about that would reduce cross-hemisphere activity in these individuals in archetype 5. And we see evidence that suggests that might be the case. So SCQ here is a measure, it's a social communication questionnaire, and it's a measure of social communication. And we see that the more genetic burden that you have in these corpus callosum genes, the higher you score on the SCQ, which higher means more impaired from a social standpoint. So the people with a lot of genetic burden, specifically in these corpus callosum genes, tend to have higher SCQ scores and are more impaired. So just to recap, from a diagnostic perspective, higher IQ, specifically in the context of autism, seems to be associated with more internalizing symptoms such as anxiety, low sense of self-worth, withdrawal, poor interpersonal relationships. And from a cognitive perspective, a processing speed and verbal comprehension discrepancy, where processing speed is significantly lower, that is associated with reduced attention, more internalizing symptoms, and more deficits in adaptive and daily living skills. So the cognitive and the diagnostic paths seem to converge on these consistent themes, especially the internalizing symptoms being something that is more specifically a feature of 2E, even relative to normal IQ ASD. And so we hypothesized that differences in brain connectivity might explain the processing speed weaknesses and the verbal comprehension strengths in 2E individuals. So this begs the question, how much should society accommodate for these discrepancies versus what might be done at the therapeutic level to boost processing speed? Now, I'm not saying that either one should be done without the other, but I think both are probably important. But it's important to recognize that both of these approaches will take a lot of effort. One thing we'd like to know is low processing speed causal for all of the behavioral and other cognitive issues that we see, or is it just simply associated? Another question is, can processing speed be improved through training? There's some evidence in the geriatric literature that processing speed can be improved through training. And so it's kind of a fascinating idea that doesn't have a lot of data to it right now, but the idea that you could train on some lower level process and potentially see higher level improvements. But again, there needs to be a lot more data on that. And then finally, are there medications that improve processing speed? I'm sure there, to be honest, that's a new question I've asked, and I'm sure there's probably some literature on that, but I at this point, I'm not really well acquainted with it, but that's something that I think is an interesting question. Okay, so now for the final stretch of my talk, I want to talk about some data that we collected through Spark. So we recruited in a follow-up study about 1,800 families in Spark. And one of the limitations of Spark is that we don't have like clinically validated IQ data in Spark. It's virtually all parent report. And so it's kind of like looking through a glass darkly. And but we used the data that we did have available to kind of enrich for high ability. And we also have exome sequencing data. That means the part of the genome that codes for protein, which many regard as the most important part of the genome. So we have the sequencing data there. And we also have array SNP or single nucleotide polymorphism genotyping in these individuals. And so we use SNPs to calculate a variety of polygenic risk scores or polygenic scores, including estimated educational attainment. Oh, and this is the same slide here. I realized I forgot to delete this one, but this is the overall, this is like the parent reported IQ bin. It's not the actual IQ score, but the bin that a parent reports to the child. And so in red, you see this is where our sample is, and then in blue is all of Spark. So a polygenic risk score, which I'm going to talk about coming up here, is when we basically ask if we take the collective effect of all of the variation in the genome and sum that together, both the good and the bad, or the positive and the negative, what effect do genes have on an individual's blank? And that could be BMI, body mass index. It could be educational attainment. It could be risk for schizophrenia. It could be your cholesterol levels. It could be a lot of different things. But the idea is this, that you draw from all over the genome, and often this is the sum of thousands or even millions of individual tiny effects. So just a quick aside on the genetics of educational attainment, it may or may not shock you that educational attainment, or years of education, which I call AE from now on, is among the most strongly genetic of complex traits. Many of the implicated genes that have been found through these very highly powered studies are the same genes discussed yesterday in Ted Abel's talk on learning and memory. And in the most recent paper that I'm showing here, they found that basically anything that is known to have a role in neuron to neuron communication has a detectable effect on educational attainment. So, but as a trait, educational attainment is some combination of raw cognitive ability combined with perseverance over a long period of time. We know it's not exactly the same thing because people have looked at this. It's not exactly the same thing as IQ. It's not exactly the same thing as a score on a cognitive test because, again, it's integrated over a long period of time. And so we need to do more to understand this trait, but it's a very widely available, widely recorded trait. The number of years of education is available in very large cohorts and turns out to be a very good proxy for a lot of things. So we can calculate a polygenic propensity score for educational attainment. Now, just to put this into context of how powerful this is, the yellow and the blue are two different out of sample cohorts that they looked in. And this was a study of several million individuals. And so what they did is they broke up the polygenic score for educational attainment by quintiles. And so in the lowest quintile, they looked at mean prevalence of college completion, and they found about 10% in the lowest quintile. And then as you can see, there's almost a linear trend as you go through the quintiles, and actually quite a substantial difference between the lowest and the highest quintile. And again, this is just purely looking at a person's genetics. You can predict college completion with that level of an effect. And then also, I wanted to point out that autism itself is genetically and positively correlated with educational attainment, as you can see here. This was from a study a couple of years ago, a genome-wide association study on autism, and they found these correlations, educational attainment positively correlated with autism risk. Okay, so back to the research match. Among a number of different things, we looked at the child behavior checklist, and we looked at the adult self-report for their parents. And so for the first analysis that I'm going to show you is we can ask what this polygenic estimate of educational attainment, what does that predict in the adult self-report, which is an inventory of self-reported indices of psychopathology. So items are rated on a 3-point scale. 0 is not true, 1 is somewhat or sometimes true, and 2 is very true or often true. And so I'm just going to show you the top positive and negative associations. All of these are nominally significant at p less than 0.05. So on the top are the positive trends. So this item is, I can do certain things better than other people, which you might expect to hear from people who have a higher polygenic propensity for educational attainment. So there's that effect. I feel that I have to be perfect is another positive association with polygenic educational attainment. This is concerning, but thinking about killing oneself is positively associated with polygenic educational attainment. And feelings of worthlessness or inferiority positively correlated with polygenic educational attainment. At the same time, the most negative associations are also things that you might expect. So there's less somatization, like headaches and aches and pains. There's a negative endorsement of I don't have much energy. So these are people that do have energy. And also the financial responsibilities. I fail to pay my debts or meet other financial responsibilities, negatively associated. So the sense of being vigilant about one's responsibilities is something that is clearly associated with polygenic educational attainment. So we also looked at another polygenic score for cognitive ability, and we found similar results. So genetic potential for high cognitive ability is linked to feeling superior and that one has to be perfect, for crying a lot and feeling lonely, for feeling overwhelmed by responsibilities and worrying about the future. Now, for the last couple minutes of my talk, I want to talk about creativity and autism and give just a few early insights into this analysis. We have a long way to go on this analysis, but I wanted to show you what we've seen so far. So we often focus solely on the deficits. I think that's less true, certainly in this conference, but in the other conferences that I go to, and I'm sure you see this too, we tend to focus almost exclusively on deficits. But here we want to understand what the perceived strengths are and their potential relationships to specific patterns of behavior. So as with the cognitive archetypes, we derived creative archetypes in autism, and then we examined their relationship with behavior and genetic risk. So I want to show you what was actually put in front of the people who participated in this study. So if a parent is reporting for their child, then this would say, I would describe blank and the child's first name as. They also did this for themselves, incidentally, but I'm not going to show that analysis. But this is for parent to child. So it starts off just neutral, but a parent can move this slider around to either positively or negatively endorse that their child is a reader, a writer, a performer, a musician, and so on. There are other things that you'll see in the next slide. So again, parent can move those around according to their perception. That's not to say that this is a gold standard measure of creativity, but it's simply a reflection of the parent's perception. So when we do the archetypal analysis, I realize this is a lot to take in here, but this is our whole sample. The first little sliver here is mixed or indeterminate, where the people in this group were not unambiguously assigned to an archetype. But you can see we have reader, writer, musician, performer, storyteller, creative type, geek, numbers person, problem solver, artist, and dancer. And orange means that there is a positive endorsement, and blue means that there was a negative endorsement of that thing. So the first is archetype 1, or what I call, I had to name all these because the computer doesn't name them for you. You kind of have to look at what's in there and come up with names. And so the first one I called the bard, because one of the ways that it differentiated itself was that it was strong positive endorsement on storyteller, creative type, and geek. So if not bard, you might think of this person, and I think we probably all met these, as the world builder. You know, like they'll have these very complex systems of fantasy worlds and stuff, and alien civilizations and all of that. I think that kind of person would fit into this category. Then A2, we have the engineer polymath, where they have pretty strong endorsement in number related things and problem solving related things, but and this is sort of in contrast to A3, which is the engineer. A3 has those things too, but basically a negative endorsement for anything outside of it. So they're not a creative type, not a storyteller, not a performer, a musician, or writer, and maybe sometimes a reader, and also not an artist, not a dancer. But so A2 is sort of like the engineer, but plus endorsement across these things that are not necessarily thought of as engineering centric. And then A4, we have the performer, where there is stronger endorsement, and this is, well, the strongest endorsement of being a dancer, but also somewhat of an artist, a musician, and a performer. And then the polymath, A5, is pretty positive endorsement across everything. Now, there may be reasons to be skeptical of whether this is actually the case, or whether it's more reflective of the parent and how they perceive their child. But we don't have a way to disentangle that with the data that we have. And so we're just calling these the polymath. Then we have the writer, where there's a specific and very strong endorsement of being a writer, but also a reader too. And then just kind of generally weaker endorsement of other things, some endorsement of being an artist. And then A7 was a little bit hard, because the thing that really defined this was not really any positive pattern, but a fairly strong negative endorsement of the geek slider. As you'll see in a minute, this is part of our sampling routine, was that we also sampled individuals that we suspected were not high performers, high academic achievers. And this group seems to be pretty strongly represented there. Okay, so we asked the question, what is the relationship of these archetypes to the parent-reported full-scale IQ, and what about patterns of behavior? Does a problem predict a basic mode of creative pursuit? So the first one, again, this is the parent-reported full-scale IQ, and the engineer polymath scores the highest on that, and the not geek scores the lowest. And I think it's interesting that the mix is just right in the middle. These are relationships between, in the columns, you see factors that were derived from the child behavior checklist, externalizing, internalizing, somatization, and so on and so forth, and then the different creative archetypes. And the one thing that I just want to point out here that I find really interesting is the strongest predictor of what kind of creative archetype you will be is this withdrawal factor. Not enjoying being around people. And there were a number of items that loaded pretty specifically on this. So the engineer archetype was very strongly positively associated with this withdrawal, as was the writer archetype. So those two archetypes very positively associated with this withdrawal factor. And then these other ones that are in blue, those are negatively associated. So the engineer polymath is unlikely to show strong signs of withdrawal. Likewise, the performer and the polymath are unlikely to show signs of withdrawal. So there are other things in there too, but for the sake of time, I'm not going to go into them. But I think this is a very interesting thing to think about, how behavior influences the manifestation of creativity. So are there patterns of genetic risk that inform the creative archetypes? So we looked at family history of different psychiatric disorders, and also we looked at common polygenic propensity scores. Again, there's a lot to take in here, but I'll just quickly scan through these. That we actually found more positive than negative associations in terms of educational attainment was very strongly and significantly associated with the engineer polymath creative archetype. And then the cognitive performance, the test-based one, was strongly associated with the bard. In terms of negative depression and depressive symptoms were, and they came out actually being negatively associated in these, except for the not geek was strongly associated with a loading for genetic risk for depression. And then I'm coming up to the end here, but this is just looking at kind of the same thing, but with family history. I think it's interesting to point out that the not geek, again, has a strong family history of intellectual disability, which is consistent with the other patterns that we saw. And the opposite is true for the writer. Okay, and this is just a bonus, but I wanted to show, we've been collaborating with John Strang at Children's National on, we do a lot of work in gender identity and the genetics of gender identity, and specifically all that within the context of autism. And so we, in a separate study, we evaluated adults, or we had them fill out surveys that helped us to figure out quantitative measures of gender identity. And that ended up in 5 factors, which I'll show you in just a second. And what we found was that when we looked at polygenic educational attainment, there was an effect specifically among those diagnosed with autism, where increased polygenic educational attainment was linked with greater diversity in gender identity. And that's what this looks like. So pay attention to the purple box here, which is ASD diagnosis and in the highest quintile of polygenic educational attainment. So here we have these factors for divergence from their binary identity, the non-binariness, the neutral style, bothness, and then genital dysphoria. And this was the only one for which there was not an association. So these findings support the increased gender diversity in autism, and further suggest that increased diversity in gender identity could be a 2E linked phenomenon. So to conclude, a better understanding of the archetypes of creative strength and their relationship to problem behavior and neurobiology and genetics may lead to more tailored accommodations, might lead to more tailored education, training, and therapeutic interventions. And I think it's remarkable that some of the creative archetypes actually showed little genetic or familial association with autism itself, but were instead associated with genetic risk for other conditions on top of the autism. So we have a lot more work to do in analyzing this data, but I'm thrilled to be able to show you the initial fruits of our collaboration. So I just want to end here by bringing up this slide again of I want you to use your imagination of thinking about how you would fit into into a collaborative network that was able to recruit these large samples so that we can get a more accurate and more robust understanding of twice exceptional phenomenology. And I want to end with a very Iowa-centric thing that if we build it, they will come. And so if we continue to care for and build these collaborations, and I think the summit is a very important part of that, we will be able to get to the point where we're thinking about large-scale science, where we move beyond the waiting room studies and move to stadium-level science so that the conclusions that we draw are robust and reproducible, and that we can take action based upon them. So I want to thank, of course, the Belin-Blank Center team for putting on this summit, and Susan specifically, and all the members of our collaboration, including Tomas and Ted, and also Lucas, who was instrumental in making that data set happen. Of course, all the Spark participants and the Simons Foundation. And with that, I'm going to end, and I would be happy to take questions now. So thanks, everybody, for your attention.